Good morning, you guys. I'm going to share my screen right away. At 6 a.m. here. Welcome to Hotel Life from the West Coast. But I wanted to go ahead and give you guys a few helpful hints and quick resources on how to get through Chapter 16. So I'm going to screen share with you guys and get you my PowerPoints and kind of get you squared away with looking at Chapter 16. All right, you guys, so the big picture here, molecular inheritance. You guys just spent all this time learning about mitosis and meiosis, and we said things very casually like, oh, in prophase, you can see all of those chromosomes duplicate and you get double the DNA. But how did we actually do that? So this chapter kind of breaks down for you the actual structure and function of DNA because that's critical, you guys. If we don't understand what the DNA actually looks like, it makes it really hard to understand this whole process of replication. That's the key word for this chapter, replication. How our DNA goes from one single unit to multiple double-stranded units. So they're going to spend about 40 slides. Yes, that is overkill, and I am going to blow through them, but make sure you read them one by one. But I want you guys to kind of understand why it's important that we know that whole backbone of DNA, the ladder, and all of that good helix structure and what it means. So you can kind of see that they're going to list a lot of names. And the reason they list every single name, you guys, is we always think of Watson and Crick. They're the guys who gave us that double helical model for DNA, but they didn't do it alone. Just like you guys know, the father of modern genetics was... awkward pause and silence for Gregor Mendel. And you also know that the father of, let's say, evolution, he was... Charles Darwin. I know you guys are saying it with me, but you guys, the reason we know those names is because they got the credit, but they weren't the ones acting alone. They were just the first ones to kind of put it into words, but they used a lot of help from other scientists along the way. So I'm going to flip through these slides, you guys, but what we're looking at right now is DNA replication. That's your key word. We're replicating. So we want to look at the structure of DNA and then the function of that replication. Okay, so you can see that we're going to have names like Morgan and Griffith. They're going to work together as early as 1928. One of the things that they did, you guys, was look at transformation. Okay, I want a light bulb, king, to go off in your head right now. Transformation is a lot of what we did last week in our labs. We used a plasmid, which is essentially, you know, the vesicle of a eukaryote cell. A plasmid is kind of a vesicle. And we inserted into the nucleus of a bacterial DNA the DNA of that fluorescin, right? That code for fluorescing from jellyfish. Transformation is when you're changing an organism's phenotype due to this new genotype that you have inserted. So we took a bacteria, we kind of inserted into it this new DNA, and we were hoping that that new bacteria that was created would display that phenotype, that fluorescin. Now, you guys, we're not talking about changing the genotype, right? As far as the structure. We still made more bacteria. We didn't go from bacteria to dinosaur. We still had a bacteria, but by changing that DNA just a little bit, we can change the phenotype. We can change what it displays. Now, unfortunately, they practiced this in mice. So we had a lot of mices go through their sacrifice to science. But what they did was they gave us the ability to see that you could still have a healthy mouse if you inserted that DNA. We could display some things. Now, there's still epistasis. We've got some environmental factors that could play into that. But we learned what DNA was through that. We had Avery, McCarthy, McLeod. They all kind of helped us go through that process of learning what DNA was. We're still just trying to figure it out at this point. So you guys, the first 40 slides, that's all about what the DNA was, how we came to understand what DNA was. So you guys blow through that at your own pace. But okay, here's the big thing, slide 20. DNA is a polymer, which means many of monomers, right? In this case, it's nucleotides. Remember back to chapter 1 in our major macromolecules. You know, proteins were made of amino acids, carbs were made of sugars. You had nucleic acids that were made of nucleotides. Remember, form equals function. Yes, every nucleotide has the same form, a nitrogenous base, a sugar, and a phosphate group. Now, DNA stands for deoxyribose, Remember, anything that ends in OSE is typically going to be a sugar. So there's your sugar. Typically, it's a 5-carbon sugar, and that phosphate group is going to be kind of what helps it glue together. The sugar and the phosphate constitute our backbone. They're what make up the backbone of the DNA. The nitrogenous base, you guys, that can be either adenine, thymine, guanine, cytosine, any of those 4, and we can put them in any sort of order. And you guys, these can be millions of nucleotides long, like 6 feet worth of nucleotides in a row. And whatever order we put that ATGC in makes up that particular molecule. So the nitrogenous base is what actually does the defining of what you are. The sugar and the phosphate, that backbone, they kind of help keep things structurally sound. Okay, so Chargaff's rules. The base composition varies between species. That makes sense. We can put them in any order, and certain orders are typical to certain species. In any species, the number of A and T bases is equal to the number of G and C. So remember what we've been saying off and on this semester. Apples grow on trees, cars go in garages. So those will always go together, kind of like puzzle pieces. A and T just fit, G and C just fit. So we're still kind of understanding it at this point. We're still in the 1900s. We're still trying to get this all squared away. But we did understand that DNA was varying between species and that A always went with T and C always went with G. And those amounts were always equal. They had to kind of balance each other. 5' to 3'. Okay, guys, there's a great video that I posted since I don't have a blackboard for you this week, but you can see that little yellow cloud is going to be your phosphate group. The blue kind of 5-carbon squat structure is going to be your sugar, and then the base is going to be everything on the right-hand side. How we designate 5' to 3', you always start with where the oxygen is and work clockwise. So start numbering your carbons. This is when we're going to start numbering our carbons. So from the oxygen on your blue kind of sugar group, go to the right, there's 1', 2', 3', 4', and then on the outside of the sugar is your 5' carbon. So when we say 5' to 3', you're kind of moving in that direction where you're going 5' carbon to 3' carbon from top to bottom. Think of it like reading a book, you guys. You read from, left to right. That's kind of the way that you're going to read your DNA. So that's what we're looking at here. Helpful video. I'm just going to keep going because I know you guys don't want an hour-long video. But through a little bit more work, Rosalind Franklin, she was huge in this. She had X-ray crystallography, and that helped her put together that DNA wasn't running 5', 3', 5', 3'. We had something really special going on, that we were kind of doing this anti-parallel and that we were making up a double helix. And that was thanks to that X-ray crystallography, because she could see that something was, making this X pattern, but she couldn't figure it out. So 5', 3' is how we're going to run. You can see that on the left-hand side. And then on the right-hand side, when they attach, we're actually going to run 3' to 5'. You still have your nitrogenous bases matching up in the middle with those weak hydrogen bonds kind of holding them together, but they kind of wrap around themselves, making that double helix structure. All right, you guys, I'm going to flip through this because you guys take some time. We're going to go over this on Monday after spring break, a little bit more detail. But the really important thing for these first 40 slides is just how we came to understand DNA. You guys, we still didn't get a really good grasp on this until the mid-1900s, when we're trying to figure out what these structures are and why they're so important. So Watson and Crick, they help to explain the Chargaff's rules, the A and the T, the C and the G, that double helix, anti-parallel, so they get the credit with really putting that into foundation and understanding. Okay, DNA replication. Remember, key word for chapter 16 is replication. We're looking at what happens when we go through interphase, right? Not just in mitosis, we can see all of those chromosomes that come together and join up, but how did we get all of that double DNA? From synthesis during interphase. So during interphase, when they go through all of that replication, typically for mitosis, right? You guys, growth and repair, things like that. We can kind of see now how that happens. So first thing we have to do is we have to unzip the DNA. We have to unzip it, right? We've got to take those base pairs and rip them apart. Then we have to build another strand on top of that. Remember how in that replication, we always said you had a parent strand and then a new strand, an original strand and then the new strand. We want both pieces of DNA to have an original copy, and then both pieces will also get that new complementary strand. So we're going to unzip it and make a complementary strand. This is considered a semi-conservative model. We're going to unzip it, keep that conserved piece, right? You're going to take that, the piece of paper that you made a copy from, we're going to keep it because we like to have originals. And then we're going to make a version off of that old model. Okay, we're going to flip through a lot of this. You guys, it's good to know, but I know you guys just want to know, what do I need to understand for the test? Okay, so replication begins on particular sites called origins of replication. You guys, if there are millions and millions of nucleotides that make up these long, incredible strands of DNA, we have to know where to start. There's not always just one start place either. We can have multiple origins of replication on one strand of DNA. As you see, hundreds, even thousands typically on eukaryotes. Replication can go in both directions, but I'm going to teach you guys that 5' to 3' is a lot easier. That's kind of the way we want to go. Think of being at the top of the steps. If you go to the 5th step and then walk down to 3', how much easier is it to start at the top and then just walk downhill? Now, if you start at the 3' end and then you have to go uphill all the way to 5, imagine that you'll have to stop every few steps if you're going all the way up. You know, the steps lead all the way up the Great Wall of China or something drastic. You guys, if you start at the bottom and go up 3' to 5', you're going to have to stop along the way, take a breath, then go again. That's what we're going to talk about when we talk about going in both directions. One way is going to be 5' to 3'. That's your leading strand. It's so easy. You're just going downhill. Whereas 3' to 5', you're walking uphill essentially, and it's going to be a little bit more complicated. All right, you guys, so you can kind of see these origins of replication. They're going to be these little bubbles. They kind of act as like a marker flag. On your mark, get, set, go, this is where I want you to start DNA replication. I'm going to flip through for you again. Okay, when we unzip it, that replication bubble ends up creating like a Y-shaped fork. So think about a jacket, right? A standard hoodie, you guys. As you unzip it from your neck and you're going to unzip your jacket, it kind of creates that Y shape, right? You've got both sides of the jacket kind of flapping open. That's going to be your replication fork. It's where we're actually going to start making the new strands. So take your hoodie, unzip it from the top, and you can see that each strand, original parent strand, is going to kind of fall away. Now we have a couple of different enzymes that are going to jump in there to get work done. Remember, enzymes typically end in what? ASE. So helicases, topoisomerases, polymerases, ligases. We're going to talk about all of these enzymes that kind of do the work of replicating our DNA. Vocab, 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 you guys. Make sure you're doing that vocab. So helicase is going to be your unzipper. Single strand binding proteins are going to bind and stabilize that single DNA. So imagine you have a hoodie, but your hoodie is 6 feet long. Some of those strands are going to start to flop together, but you want to make sure they don't flop back on each other and start zipping themselves up again. So those single strand binding proteins are just going to act to kind of stabilize that single strand so that it doesn't flop around and start binding on itself again. Topoisomerases relieve the strain of twisting on the double helix by rejoining those DNA strands at the end. Definitely watch the videos I posted, you guys. So topoisomerase, it's stabilizing everything. You can see we've got one strand that's going 5', 3', one on the bottom going 3', 5'. RNA primer, right? What do you put primer down to do? A paint primer is what you put down first. It's what's going to start that replication. So RNA primer will jump in. You guys, RNA is going to be what creates that next DNA unit. And we'll talk about that in the next chapter in a little bit more detail. But there's helicase unzipping, kind of winding as it's going. The single strand binding proteins are going to mark that strand, that single strand, for a little bit more stability. And then we're going to start building in that 5' to 3' direction, the new piece of DNA. All right, initial nucleotide that starts it all, primer, synthesized by the enzyme primase. There we go. Enzymes speed up reactions. Primates can start an RNA chain from scratch using that parental DNA chain. The primer is short and the 3' end serves as the starting point. So we're going to start on the 3' end, but remember we're working 5' to 3' because they're anti-parallel. All right, DNA polymerase is going to catalyze the synthesis of new DNA at the replication fork. Usually it requires that template to kind of work with. We work in about 500 nucleotides per second in bacteria and 50 per second in human cells. Nucleotides, so every one of those bases, you guys, ATGC, 500 a second in bacteria and about 50 in humans. We're going to learn in the next chapter that we typically work in chunks of 3, but just for the purposes of replication, that's a pretty quick flow. All right, as we add that new base, right, we're providing a little bit of ATP because when you're building something, when something is anabolic, we're going to have to require a little bit of energy. It's endergonic. We need a little help. So this is where ATP is so critically important, you guys. We need a little bit of ATP. In particular, this is going to be DATP, which has a little bit of deoxyribose, and regular ATP has ribose. The only reason that's important is because it just varies on to which strand it'll be powering. All right, so here you can kind of see on slide 68, you guys, what we're doing. We've unzipped it and we're gonna build with DNA polymerase. We're gonna add that other phosphate group and attach it to that other strand. The parent strand is going to get a new template strand. All right, so DNA polymerase. Only to the free 3' end of a growing strand is where we're going to add. A new DNA strand can elongate only in the 5' to 3'. So remember, you guys, that 3' end means that it has to link up with a 5'. We work 5' to 3'. Start at the top of the stairs and just walk down. It's so much easier. So if you see on slide 70, 5', 3', we can see the direction that that's moving. It's continuous elongation. You don't have to stop. We look at that 3' end on the right and we just start building, working right to left this time. We're going to kind of move right to left because we've unzipped it and now we need to kind of work our way across. So 5', we're just walking down the stairs and we're going to keep building to the 3' end. It's continuous. There's no interruption. It's very easy to walk down steps. Now, what we have to consider is that there may be another strand that's going to be a little bit more challenging. That's your lagging strand. So what we need to look at is our lagging strand. The leading strand is the one that we can just find the 3' end, start at our 5' end and work 5' to 3', but not both strands are going to work like that. The one that goes the opposite direction is called our lagging strand. This time DNA polymerase must work in the opposite direction, away from the replication fork. So we're kind of going uphill this time, you guys. Now we're going to create Okazaki fragments and we're going to glue them together. So instead of doing it in one long continuous strand in the direction towards the replication fork, we're going to go in the other direction. We're going to make little chunks until we have to stop, take a breath, and then we're going to glue them all together. This is a really complicated picture. You guys, a picture is worth 1,000 words. Just go step by step, right? 1, the primase makes RNA primer. 2, DNA polymerase 3, which is just another one of our enzymes, will make Okazaki fragments. So this time we're going to make short little chunks of DNA replication, right? Little small fragments. We're going to put those fragments together by using ligase. Ligase is going to be kind of our gluer. He's going to go behind everything and glue it. Definitely watch the 2 videos I posted that help put that into animation. Your book will give you like 6 different slides on the same process, you guys. But here's an example. Slide 87 is great. It's showing the big picture. Our double-stranded anti-parallel DNA, semi-conservative. We're going to keep one and make a new one. We're going to unzip it and we're going to replicate towards the replication fork. So start on the right-hand side of the slide and work your way towards that replication fork. Your leading strand is on top because it ends with the 3', so how easy is it to just jump 5' and just start working in one continuous motion to remake that new strand of DNA. However, at the replication fork, you can see that we also have to do the lagging strand. So we'll start over here. We're going to start with the RNA primer. We're going to have a primase. And then we're going to have DNA polymerase 3 come in and make these little Okazaki fragments. You can see the ligase at the end is going to come by and just glue everything together for really strong hold. So it's still structurally as sound as our leading strand. But hopefully you're starting to wonder if it's always as nice on that lagging strand as it is on the leading strand with no errors in that replication. The big take-home message, you guys, is that bacterial DNA replication proteins and their functions are very similar to eukaryotic and some human and animal. This is the table. Table 16.1 is going to be the key table for all of our different proteins and enzymes. Remember, enzymes are proteins. Helicase is going to unwind. Single-stranding binding proteins are just going to kind of stabilize those strands, leading and lagging, to make sure that there's no issues. Topoisomerase kind of relieves overwinding strain ahead of that replication fork. It's going to kind of work to stabilize in front of our replication fork. Primase synthesizes, makes an RNA primer at the 5' end of the leading strand, and then each Okazaki fragment on that lagging strand. DNA polymerase 1 and 3 are going to be enzymes that kind of help at different points. DNA polymerase 3 acts as a template, synthesizing new DNA by adding nucleotides. DNA polymerase 1 removes those RNA nucleotides and replaces them with DNA. We're going to make this make sense, you guys. Remember, RNA doesn't have the ATGC that our DNA does, so it kind of works in different little segments. But for this class and this chapter, for right now, just focus on the fact that we're just replicating DNA, so don't worry about that just yet. We're going to have DNA ligase. Ligase is your glue. It joins those Okazaki fragments together. And then your text is going to go into more detail on that. Okay. Think about this in terms of your cookbook, you guys. Think back to when we started talking about the nucleus, held that information, the copy of the cookbook for your grandma's secret recipes, right? We call that the code for life. DNA is your code for life. We have this replication machine. A DNA replication complex is just going to be a situation where all of your different enzymes and proteins, all the proteins that are needed to start this process, to come together to make a big machine, a complex. It can be stationary during the whole process of DNA replication, or it can kind of pull it in, reel it in, that parental DNA. We still don't have a good idea of what this is, but we know that these all have to come together in order to make this work properly. All of these enzymes, proteins, they have to be working smoothly as this unit, as this complex. Overall, you guys, all we're doing is trying to make new DNA from our original parent template. Now, proofreading and repairing. DNA polymerase is like Bob the Builder, right? DNA polymerase is what goes in. He's got a tool belt. He's going to go in and add all of these new nucleotide bases and put them together. But he also has the incredible ability, think of Bob the Builder with magic glasses, right? He has the ability to repair any damage and make sure that no errors occur. Now, his job is affected by environmental factors like X-rays, UV exposure, but for the most part, there are very few errors in just DNA replication because DNA polymerase is so good at proofreading that newly made DNA and correcting any errors before it continues on through mitosis. So, in a nucleotide excision repair, a nuclease, an enzyme, will cut out and replace damaged stretches of DNA. So as DNA polymerase, Bob the Builder, is working along and proofreading everything and building it up, if there is anything that's wrong, they can cut out those segments and replace them. You can kind of see an example on figure 97 or slide 97. If there is an error, something doesn't link up right, a nuclease will come in, DNA polymerase will resolve it, ligase will glue it back together again. It's not a foolproof system. The error rate is pretty low, but it's not zero. This is all still happening during interphase, you guys, in that S phase where we're synthesizing new DNA. Sequence changes may become permanent. They can pass to the next generation because if we do replicate poor DNA or error DNA, it will go through mitosis once, twice, hundreds of times. If it's during meiosis, we could actually end up passing that repair damage to an offspring or a gamete, if you will. So it's not zero, but it's very, very low. DNA polymerase is like Superman. The fact that he would miss something would be unheard of. Typically, he can catch most errors in that DNA replication. All right, you guys, the rest of the slides are all just kind of going back through what we were talking about. It's going to talk a little bit about histones, because histones are what kind of bind together a lot of those chromosomes into chromatin. I'm going to post a couple slides that I think are really helpful in our file folder to help you kind of understand these histones. But just know that, you guys, once we've replicated all this DNA, we've got a nucleus full of just mass amounts of DNA that's been replicated. What we need to do is kind of put it in some sort of order, right? You've got to pack it in your suitcase. What we need to do is make it chromatin. So what we do is we have these proteins called histones that kind of package it together. There's a really short 3-minute video under the weekly folder that shows how fast this happens. But the histones kind of come in, you guys, and they take all of this newly replicated DNA and they put it into an order, right? They're going to put it in this packing system. Unfolded chromatin just looks like a string with beads on it. So you've got a chromosome, you've put these histones all over it, these proteins, and it would be like making a friendship bracelet when you're a kid. You've just slid some beads on a piece of string. This kind of look is going to be a nucleosome. It's going to be a piece of DNA with histone on it. Now, they're going to have 4 basic histone types. They're going to wrap around. I don't want you guys to worry about this for the test, N-terminal tails and nucleosomes and all that good stuff. Just know that what we're doing is trying to condense it. We've got all this new DNA that's just been replicated. We're going to add some histones in this nucleosome kind of structure, and then we're going to continue and continue and continue to tighten it till we get to the chromatid, sister chromatid level. So that's all you really need to know for this, is that histones are what are going to be what come along and help take all this newly replicated DNA and make it into something that we can actually organize and linearly fashioned, put together on a metaphase plate and pull apart appropriately. So just think about it in terms like that, right? You've got all this DNA and now we need to get out of the nucleus, which is going to be critical. We've got to break down that nuclear envelope. Why did we break down the nuclear envelope during mitosis and meiosis, you guys? We've got to be able to attach those spindle fibers. Spindle fibers have to kind of lasso. They're going to whip their lasso and grab that centromere on the chromosome. We need to have the ability to do that. So we need to put them in a more condensed chromatid structure and match them up with their sisters. That's the whole job of histones. And we're going to have a couple type of histones, you guys. We've got euhistones. There's a couple different versions, but for the most part, just know histones are what help us put that DNA into a more tightly packaged structure. This whole chapter is just about replicating DNA. So here's what I mentioned before. Loosely packaged chromatin is euchromatin. Interphase, a few regions of that chromatin get more and more highly condensed, which is heterochromatin. Dense packaging of the heterochromatin makes it difficult for the cell to express genetic information. This is how we're going to cut on and off the ability of these pieces of DNA to go through these processes. If it's loosely packed, we can go through DNA replication really easy, and we're going to talk about that next chapter. But if we don't want it to go through any more of this, transcription and translation that we're going to talk about, if we highly pack it together, it's going to make it difficult for genetic information to code. So we're not going to have the ability to do that. Histones can undergo chemical modifications. You guys remember what all of this is just atoms that are being held together in very specific shapes. So if you change the pH, the temperature, the chemical nature, that can change the shape of these histones because they're just proteins, right? They're proteins. Proteins, when they bind to stuff, they change shape. When they come around temperature and pH, they also change shape, which can affect how they display their expressions phenotypically or genotypically. The rest of the slides are just some examples that your text will talk about. We're going to go over this in so much more detail, you guys. I know this was a quick crash course, but I didn't want to make this too long. I'm going to post a ton of videos. They'll be short. They'll be animated. Since I don't have a board, you guys will want to see some of this in action. But for the most part, you guys, DNA replication is just how we're going to make that DNA make a copy of itself during interphase, synthesis phase, before we go through mitosis and meiosis. We're going to unzip that double-stranded helix DNA. We're going to kind of use DNA polymerase to build new strands off of the original parent strands. This is called semi-conservative, right? We're keeping half. We're going to use DNA polymerase to proofread, make sure everything's okay. We're going to use helicase as the zipper to unzip it. Because remember, those hydrogen bonds are weak, but we still need something to kind of go through and break the bonds between those atoms and the shared electrons. 5', 3', we're going to go over that in so much more detail, but just remember, typically we like to work 5' to 3'. It's easier to walk down steps, so start at the 5th step and work your way down to the 3rd, but we can go upstairs, right? We can go uphill. We can go 3' to 5', but we're going to end up with Okazaki fragments that we need to glue together with a little bit of ligase. Watch the videos I've posted. Make sure you don't have any questions. You guys, I will still be here if you have any questions or concerns. I'll post a couple more videos, email me, and then we'll start chapter 17 tomorrow. Have a great rest of your week.